Right. First, I'd like to thank you to the organizers, Phil and Raphael, for giving me this very nice opportunity for me to talk about my work. And I really like 3D-seq. This is really a precious meeting talking about three-dimensional protein bioinformatics. Today I talk about our new direction of research. Well, you know, big data has been there for many years now. But when people talk about big data in biology, usually it is about next-generation sequencing, RNA-seq, so many sequence information, and expression data, including RNA-seq, or some network, protein-protein interaction network, gene expression network, or drug-disease network, and so on. And also, recently, there are clinical data available, and those consist big data in biology. Well, often I feel, where is the three-dimensional protein structures? I love to work on protein structures, but often I felt that big data is not covering much in a structure aspect of proteins. However, here I would like to claim that there are still new things going on in the structural data, and there are new field we can work on. Obviously, the PDBe is the source of our structural information. It's now have more than 15,000, more than actually 150,000 structures available. And also, some of the speakers this morning has talked about, the structure is now, there are proteins which do not have function information. Now, recently, we have observed lots of low-resolution structures coming from electron microscope, also electron tomography. Those are very different type of structure information compared to the X-ray structures or NMR structures of proteins and biomolecules. Also, as we see in some of the talks this morning, now it is possible to generate lots of, predict lots of protein structures. Now, but the protein structures have errors in it. So we may want to consider handle the predicted structure slightly differently from the crystal structures of PDBe. Also, protein is not a rigid structure. It has a dynamics, implicit dynamics in it. So this data is huge. Well, there are already a database of molecular dynamic simulations, but these are not much explored mainly from structural bioinformatics point of view. And unlike 15 years ago, now we have the drug data. Traditionally, the drug data, compound data are not available freely to academia, but nowadays there's a nice database, PubChem, and it has a huge number of compounds and activity data available. Now, in this talk, I will talk about what our group has been doing using the electron microscope data. This is Titan Cryos. This is one of the state-of-the-art electron microscope available in the market. And let me just quickly go through how the structure is determined from electron microscope data. First, the samples are put into the grid, and the proteins will be determined in 2D in a different orientation. So actually, there was a huge computation problem to develop the 3D structures from 2D structures, because 2D structures are in a different orientation. You need to cluster it, and then you need to align it. And actually, there was a motion also. There was an intrinsic motion of the proteins, and that needs to be canceled out. So there was a recent very breakthrough of the computational methods of how to determine these 3D structures from the 2D map. Now, once this is 3D map is made available, this is a 7 angstrom resolution GROW-EL structure. Now, from this data, so 3D data, we can work on using traditional structural bioinformatics techniques. Well, the primary interest is to develop the 3D structures from this EM map. If the homologous structures are already solved, or the structure of the protein has been already solved, the problem is mapping those structures into the EM map and doing the rigid fitting or doing some flexible fitting. There is some still techniques not satisfactorily developed in the flexible fitting area. But if the structures are not available for fitting, then you need to look at the resolution, the density, and then identify the secondary structure elements, and then also trace the main chain, and then make the model. And here, so in the 3D structural bioinformatics, I would like to propose that this is an area, and this is an area we can really work on. Now, the counterpart of PDBe, which EM data is contained, is called EMDB. In the okay, let's see the EMDB statistics. Now the x-axis is the year, and y-axis is the number of entries. Now it's reaching about 5,000 EM maps available in EMDB, and about 1/3 of the EM map has the fitted PDBe structures to the map. And so this is showing how the resolution is improving over years. X-axis is again the year, y-axis is the best resolution, in the smallest resolution achieved at that time. In 2002, it was around 10 angstrom, but recently it's going down to below 2.5 angstrom. This is called near atomic resolution. However, if you look at the statistics of different resolutions, maps of different resolutions, again, x-axis is year, y-axis is a number of entries. It is true that high-resolution structures less than 4 angstrom is increasing, but also medium to low-resolution EM maps are also increasing. So when we deal with the EM map, it's slightly different from how you deal with the structures from PDBe. You need to deal with the structures of relatively high resolution and also low resolution, and then do the bioinformatics using that various type of data. So the 4 topics I cover in this talk is, first, we have worked on very basic functionality of the database, which is the search. And also, we recently examined how the structures are variable, how the structures, what the quality of the structure is relative to the resolution of EM map in EMDB. And past years, we have also developed the methods for fitting the structures to EM map for low resolution maps. And most recently, we are working on the main chain tracing methods in case that EM map has a higher resolution below around 4 angstrom. Okay, so here is the search page of EMDB. Now, what you can do in EMDB here, it's similar to PDBe, is a text search. So you can search the EM maps by the name of the authors or name of the proteins or the organisms and those kind of text information. But in EM maps, unlike a structural comparison method like CE, DALI, or TM-align, structural comparison methods, EM map comparison method based on the structure information is still not well developed. So I would think that the search methods is a very essential functionality for a database, but in EMDB, well, if you wanted to search a map which has a similar structure, similar isosurface structures in the database, there's no methods to do that. And a couple of years ago, we have developed methods for doing the isosurface-based EMDB search. It's called EMSurfer. Actually, we have used that technique also for the same technique for a very quick PDB database search, but let me explain how this method works. Okay, it is also linked from the EM Data Bank website. And okay, what it does is, so it compares the query EM map, or maybe a simulated EM map against the EMDB. In real time, you submit the EM map, or you select one of the EM map in the EMDB, and it does a search instantly. And then, yeah, just like BLAST, it will rank and quantify the shape similarity of your query against the EMDB maps. And actually, what it means is that it provides you the metric of the shape similarity between the query and then EM maps in the database, which is very fundamental in the fundamental aspect of this work. And then, well, if you have several EM maps of different states, biological states, which is different somewhat by eyes, this method will quantify the difference between the maps. The method used, the main method used in this search is called 3D Zernike descriptor. What this Zernike descriptor, this 3D Zernike descriptor is the series function of the series expansion of the three-dimensional function. It has the spherical harmonics in it, and in addition to that, it also has a radial function. If you use the spherical harmonics, actually you can only describe a shape which has only the one surface along the one direction, but because it has the radial function, very complicated shapes which have multiple layers of the surface along one direction can be also represented. What it does is, okay, so, and then the base function, this is a base function, and the base function there's a series of base function which is determined by the parameters n, l, and m. Now, fx is the function which describes the 3D shape of the EM map or protein's 3D structures, and by taking the moment, you will have one parameter here, coefficient values, and by computing the series function, you get the vectors of the coefficient values of the 3D Zernike descriptors. A nice thing about it can be computed in the rotation invariant way by taking a norm out of for one of the parameter m. So because it is a rotation invariant, once the EM map is described by the 3D Zernike descriptors, it is described as a vector, and you don't need to rotate it or translate it for comparing the two EM maps. So it can allow very quick search against a database. So in the EMSurfer, given a raw EM map, first we construct identify the control level. In the EMDB, usually the author recommends a particular control level. So according to the control level, we make the shape, isosurface shape, and in addition to that, we also make much higher dense regions to capture the core of the protein, core of the biomolecules, and then for each of them, we compute the 3D Zernike descriptors, series expansion, and then compute the coefficient values and concatenate them. And that's how each EM map is represented and stored in the EMSurfer. In the EMSurfer, the data is updated every week. Well, if you have, for example, a PDB structures, and if you want to search EM map, you can simulate the EM map of the PDBe structures and do the search instantly. This is, okay, so this is one example. From the 3D Zenliku descriptors, we can also compute the surface structure back. So this is the original map of the KEYB, KEYC complex. This is a relatively low resolution, 16 angstrom. And because this is a series expansion, if I cut the series expansion at the earlier level, then it is representing very low resolution structures. And as I extend the series expansion, I can describe the structures more and more. So in one sense, I can also control what kind of, what level of structures I'm representing by the 3D Zenliku descriptors. And EM surfer, you can, if you just wanted to search, you can put the EMDB ID. In EMDB, the EM map has 4-digit IDs. Or if you would like, you also upload the EM map, and then it will be converted to the 3D Zenliku descriptors, and then search against the whole EM maps in the EMDB. This is one example. This is the query. The first one is a query. This is 30S ribosome structures. And then those are the top hits from the EM map. This is another example. This is a kinesin. Kinesin is actually tubular structures, and it has an elongated, this is just one unit of the map, but it can, for this kinesin, you can also find the related entries in the top hits. And then this Euclidean distance is a distance between the between the two shapes. Empirically speaking, if this value is less than 10, usually those are the biologically related molecules. Well, in the EMDB, we don't have CATH database or a SCOP database. So because the number of entries are small and, still small, but still, now it's getting 5,000. So but the EBI EMDB team has a data set of the four type of the EM maps: eukaryotic and prokaryotic, ribosome, chaperone, and virus. And then we did the search test to see if we can from each of the query, if we can get the EM map of the same type in the top hits or not. Well, this is how the results look like. About 80% of the cases, the top hits include the structures of the same type. Well, since this data set is relatively small, we want to expand this benchmark data set, but this needs a little bit manual curation. that takes us a little bit time for doing that. This example is showing the how the structure similarity is quantified for related EM maps. In this paper by Caroni et al., they solved the structures of hexameric triple A plus chaperone, CLPB. This is CLPB is here. This is one subunit, and it makes a hexamer. And they solved this EM map with CLPB, hexamer of CLPB, with the protease called CLPP. And there are 9 entries. These are the 9 entries. Well, the description, as you see in the description, there are some differences in description. The distance is shown here is a distance from the top entry. Some of them are within 10, which is how it's supposed to be. But there are some entries which are very large values. And we are wondering why those entries are supposed to be from the same protein complexes, but still have a very large number. And actually, it is capturing really the different biological states. Well, I cannot go into the detail, but this is a query. And then, for example, the one which are very large value, or large distance was this entry. This for the first part, CLPB part is the same, but in a different biological states. And then there is a CLPP missing here, and that's why the distance was very, very large. So similarly, visually different the shapes of the structures it can be quantified by the numbers. So I can, for example, compute and do the clustering or draw a dendrogram to visualize the structural similarity of EM maps. Okay, now let's go back to the resolution histogram of the EM map resolution. As I mentioned before, some of the maps are very high resolution within 4 angstrom, and some of them are above 15 angstrom. But the problem here is that, one side of the EM map have the PDB structures fit into the EM map. However, it's not very clear how the structure is validated. This is what the EMDB the website says. How are coordinates derived from EM reconstructions validated? Well, it says it's by visual inspection. Actually, there is no way to really validate how if the deposited PDB structures is really fitting to the EM map or not. For EM map, there is a way to compute the resolution of the EM map, and then there is a way to validate if the EM map itself. And then, of course, once you have the protein structures in PDB format, those PDB structures are validated by checking the bond angles and the bond distance and so on. But the fitness of the structures to EM map there's no way to really address the fitness of the structures. So we wanted to test how well actually the structures determined by EM is supported by the EM map. This is published on Structures early this year. So what we did is first we made a data set of 49 maps and PDB pairs. The way we did it, we started from the all EM maps on EMDB and then obtained the all EM maps, obtained the EM maps with the PDB structures fitted on it. And then in the case that the structure has a symmetrical assembly, then we symmetrically assemble the structures. And then we map the structures to PDB. This mapping is provided from in EMDB. And then we checked, then we filtered out a couple of pairs of PDB entries and EM map. If an EM map in PDB structures is only fitting a part of the EM map, then also remove the DNA structures. And that consists of 49 maps and PDB pairs. From these, the map PDB pairs, what we try to do is to further refine the structures by ourselves, starting from the deposited structures in PDB and EMDB. We use two methods. One is called MDFF, Molecular Dynamics with Flexible Fitting, developed by Klaus Schulten's lab. And another method is Rosetta, nowadays frequently used for structural determination from EMDB, from EM map structures. So, so for individually, we refined the structure by MDFF and Rosetta, and then compared, actually, first we compared if our refinement or like a modification of the structure ever changes the already deposited structures from PDB or not. And then at last, we also compared the refined structures by MDFF and Rosetta, and then see if there was any discrepancy or not. This is how the MDFF works on the left side. MDFF is a molecular dynamic simulation, but it has a EM map fitting term, which is basically driving driving a structure to the high dense regions of the map. In Rosetta does a similar thing, but it uses a slight different function. This is a cross correlation between the density of EM map from experiment and also computed density of the protein structures in the model. This P0, PO is an experimental map, and then PC is a map of the simulated map of the structures, and then it is just simply computing the correlation. So the protein should go to the dense region of the protein. And because it's Rosetta, it does a segment. It identifies as the low dense regions of the structures, and then change the fragments, and then do this Rosetta fragment-based refinement to get the final results. So the approach is, of course, both very reasonable, but have a different flavor in the algorithm. Now, well, first let's see how the structure look like, how the refined structure look like. This is an example of relatively high resolution EM map, 3.8, determined by at 3.8 angstrom. This is a virus L protein. So the initial structure deposited by author is in cyan. Blue is the results of the MDFF refinement, and red is a Rosetta refinement. At the 3.8 angstrom level, you see some difference in the loop regions, but the differences from the initial structure, deposited structure, to the refined structures are at the loop regions, mostly loop region, and for the other part of the alpha helices or beta strand regions, we don't see significant changes. Now I go to the medium resolution structures. This is a 10 angstrom structure. At the 10 angstrom structure, we started to see some significant changes at the loop region or maybe in the alpha helical regions like these regions. So we see that the secondary structure start to shift at the 10 angstrom. Now at 25 angstrom structure, this is aquaporin calmodulin complex, and then this is done by not cryo-EM, but this is stained EM. The structure is stained, and this is done by rigid fitting. So actually, because the resolution is low, the authors did not try to refine the structures, which is very reasonable. But when you see the PDBe structures associated with this EM map, you don't, well, unless you pay attention to the resolution, you don't see, because you don't see how the structure is derived unless you read the paper, because structure looks very nice because it has been determined by X-ray crystallography prior to this EM maps. So at this resolution, obviously you see the domain motions, and actually one of the alpha helices was almost outside of the EM map. So if you run the refinement structure, refinement model, refinement methods, then this will be pushed into the envelope. So you see the significant changes in the structures if you run the MDFF or Rosetta refinement. So there was 39 maps, 49 maps, and then this is just showing that after the refinement, the left side is the MDFF, x-axis showing the cross correlation, how much the cross correlation is checking how the structure is fitted to the EM map. Positive value means that after refinement, the structure is fitting well better to the map. Y-axis is showing how much the energy of the MDFF decreased, became better. So most of the cases, the structures improved both in cross correlation and on the energy. Same thing for Rosetta. So this is just showing that almost all the cases, the refinement protocol is working okay. This is the same thing, but now I use mole probability. Mole probability is checking the geometrical the aspect of the structures, the atom clashes and then bond angles and so on. So in terms of this, outside the evaluation scoring function, it is also, after the refinement, most of the structures is improving the cross-correlation and improving actually the mode probability score. So the refinement is, here I'm just claiming that the refinement protocol is working fine. And okay, so this is how the overall structure changed on the y-axis relative to the EM-map resolution. For the MDFF on the left side, there are parameters G-scale. G-scale is controlling how much I weight the fitting to the map relative to the other MD-based terms. So as you see, well, naturally, if you see if a map has a lower resolution, then RMSD goes up. So the refinement protocol moves the structures more. At, let's say, 10 angstrom structure, 10 angstrom maps, on average, global RMSD, in terms of global RMSD, structure moves around 3 angstrom. So about roughly speaking, 1/3 of the resolution will move, well, the RMSD of 1/3 of the resolution will be changed by the refinement protocol. Same thing for Rosetta. Well, actually, in Rosetta, some of the structure moves much more than the MDFF. So the actually y-axis value and the top value is slightly different, but the same thing. At 10 angstrom, on average, the structure moves by like a 3 angstrom. This is same situation if you look at the local density. Here, there is a method called ResMap, which compute the local density of the EM map. The x-axis, the map local resolution, y-axis is telling how much C alpha positions has moved by the refinement protocol. It is moving, so actually, again, same thing, but actually it's slightly a little bit more than the global RMSD. At 10 angstrom, on average, the C alpha position moves about 4 angstrom. Rosetta is the same thing. Rosetta moves a little bit more aggressively, but basically it's the same thing. Now, okay, then the question is that, why is that? So, well, the author did not refine the structure satisfactorily or not. Well, to answer that question, we now compare the model refined by the MDFF and then refined by Rosetta. And actually, we see the same trend. At 10 angstrom, global RMSD moves at about 3 angstrom, which means that there is EM map does not have the structure information there. So Rosetta and the MDFF does not agree. Well, this is from what we learned from this analysis. So although in the EMDB, most of the cases, 1/3 of the cases, structure is mapped to the EM map. The variability or reliability of the structures really differs based on the resolution. And if we want to deal with the structures from EM map, this is something we need to think about. Well, now I show you 2 methods we have developed in the past few years. The first one is developed in 2012. At that time, the EM maps, the resolution has not improved a lot at that time. So we assumed that most of the EM map is about 10 to 20 angstrom. For that, we can do the rigid body docking. So the problem setting is that suppose I have low resolution EM map at about 10 to 15 angstrom, and here assumption is that suppose I have the structures of individual components, which is solved by X-ray crystallography, or maybe the modeled nicely by the structure prediction methods, then I want to fit these structures into this EM map. The way I do it, we did it at that time, is, okay, because we have the 3D Zelinsky descriptors, which can quantify the shape similarity. First, we dock these 4 structures by our own multimeric protein docking methods, and I'll show you in the next slides, and make that about 200 complex models. And then for each of them, I simulate the EM map, and then for each of them, I compute the 3D Zen-link descriptors, and compare the 3D Zen-link descriptors to the 3D Zen-link descriptors of the map itself. And then find the model which fit best which have the similar shape to the EM map. Well, this is how the multiple docking method works in our software suite. It's called Multi-Lizard. Multi-Lizard docks multiple proteins into a complex. For example, if you have 4 chains, and if you don't know which one is interacting, I do the docking for every pair, and then make the library of pairwise interactions, and then later, by genetic algorithm, we assemble those the pairwise docking conformations into the final model, and that was used in the EM lizard in the previous slides. In the so in the EM lizard, well, this is the results of the fitted results to the simulated EM map at 10 angstrom resolution. So there's 3 examples shown here. X-axis is RMSD of the model. Y-axis is a similarity between the surface of the model to the resolution to the EM map. Well, naturally, if the shape is very similar, then RMSD is very small. So I can, out of the 50 model, out of the about 200 models I generate, I know if the 200 models include the good one, which can fit which have the similar shape to the EM map or not. If I don't have a good model, then I can redo the genetic algorithm to make further models. In the last part, I'll show you our recent work on the MAINchain tracing methods. This MAINchain tracing methods called MAINMAST will consider high-resolution EM map of about 4 angstrom, because now we realize that now EM methods can generate lots of high-resolution structures. Now we have the EM map at around 4 angstrom, and the goal is not by structure fitting of known structures, but by tracing the dense region of the EM map, we want to generate the main chain structures of the protein. It's called de novo modeling because I don't use the templates from PDBe, but I only use the EM maps. Actually, I even don't use the fragment information like Rosetta does, and it's fully automated because we generate lots of models, but there is no need for visual inspection and try and error, which is usually done by the structural biologist. So this is the outline of how MAINMAST work. First, we have the EM map, and the EM map has high dense regions, and also these blue regions are the low dense regions. Okay, in this example, this EM map is solved at 2.9 angstrom. This is very high resolution. Nowadays, there are also much higher resolution structures deposited to EMDB. But so I use this as an example to explain how it works. Now, okay, starting from this EM map, first so this is EM map has a format of grid, and each 3D grid, the density values are mapped onto each of the grid. First, we identify the grid point which have a very dense density values. Then now, at the very beginning, there are lots of points which have, the density values above the cutoff. And then I started to shift the cluster and shift the very dense points, local dense points, by using the mean shift algorithm. What mean shift algorithm is trying to do is it is updating the position of the each point's dense region by actually using by computing the weighted sum of the previous points. The phi is the density values of the point at xn, and then this k is basically just the distance between the point y and the xn I'm looking at. So well, in principle, it is just shifting the dense regions so that the points are merged, and then the number of clusters getting to the some reasonable number of points. After I get all those points, then now I connect those points. To connect these points, we use a minimum spanning tree. Minimum spanning tree it seems to be a good algorithm to do it, because usually I still have lots of dense regions, and then connecting it to the neighbors will result in the tree structures. Now from these tree structures, okay, this is only looking at the tree structures. It is not it needs to be further refined, because like if you see these branch points are caused by the sidechains. And sometimes in the beta strand, sidechain and sidechains are connected, and the tree structures are actually, it's not like a one single chain like proteins. And then now from here, we started to refine the tree structures. Oh, okay. Well, this is showing the longest path in the tree. But unfortunately, most of the cases, longest path does not correspond to the main chain of the protein structures. So we want to refine the tree structures. Actually, not really refine it. We want to generate lots and lots of more different different trees, so that we can play with it later. It's similar ideas of generating lots of structures in the ab initio structure prediction. So okay, so this is after refinement at 20 steps. Refinement is done by taboo search. Taboo search is trying to do the conformational search, so that the next structure generated will be different from previous one. The location of the initial structures is considered as taboo regions, and then the next generation of the search will not try to go into there. Okay, at 50 steps, in this example, the structures found some density here, so that's elongated, and now further elongated, and then compared to the previous points, actually connection is also changed. Previously, this region was connected, but now this was cut here, and then start and end was now changed. And then this is, now it's further elongated. And this is actually, again, the connection is changed. It's not clear which one is correct, but this is just trying to generate lots of different models. And finally, after generating about a couple of thousand structures, then we fit the sequence to the longest path of each tree. We do it by DP, which is very familiar in threading. Because we can see the dense regions of the points along the tree, the branches, we run a dynamic programming algorithm by trying to match the amino acid volumes to the dense regions. And then this is how the map how the structure look like. And in this example, so the first model, and then the models are scored by the threading score. And in this example, RMSD is 2.5 angstrom. This is some of the examples for simulated EM map. Simulated EM map is relatively easy to use because there is not much noise in it, and then the density of the whole map is kind of uniform. There's no low dense regions or high dense regions. This is a map. this is a model, this is a native structures, this, well, alpha helical structures and beta structures, and this is RMSD of the model. And this is how the RMSD and the threading score correlates. Well, in general, there is some correlation. You see that there are a bunch of, there are lots of structures with a similar RMSD with different threading score. This is happening because of the shifting of the sequence mapped to the tree. So the structures do not change that much, but the threading score change a lot. And now there are two methods which could do the de novo modeling. One is called pathworking. Pathworking is done by the Baylor people at Baylor College, and this is showing the simulated maps, results of the simulated maps from the data set of 40 simulated maps used by their papers. This x-axis is a MAINMAST RMSD, y-axis is a pathworking method. There are two versions of it. Well, there are RMSD values, but in our case, our RMSD is around 2 angstrom. Now this is comparing with Rosetta. So the first graph here is RMSD value of the top 1 model and top 10 model. And this line, well, because the x and y-axis scale is different, this line is the y equal to x line. So actually, well, if you don't manually curate it, Rosetta often fail to fit the fragment, and that makes the RMSD value very large. But MAINMAST always connect everything, so we don't observe very large RMSD value like Rosetta did. This is a coverage. So this is computing the percentage of amino acids or fraction of amino acids within 3 angstrom to the native structures. Well, in this simulated map, our method is doing very well. In Rosetta, well, there are lots of good models, but occasionally they have a very not very good models. Now, this is the results on the experimental map. This is 30 experimental map. Again, now we only compared with Rosetta the same. Well, so x-axis is RMSD of the MAINMAST, from the y-axis is from Rosetta. Well, so if we see that the points are larger above this line, that means that MAINMAST is doing well. So, well, actually, majority of the cases, MAINMAST is doing well. This is again counting the percentage of amino acids which is within 3 angstrom. MAINMAST is always getting 70% or higher. Well, sometimes Rosetta fails to do the fragment mapping, and that makes this low coverage. This is some of the examples. Well, this is the 2.4 angstrom structures. Rosetta, well, for Rosetta, we have 2 methods. One is a default setting, and we find a better setting for Rosetta, and that's slightly improved results is given by that the parameter setting. This is another example. Okay, here this is showing actually the green. Well, green is a threading result. And then we also run MDFF to further refine it. That's where the, actually opposite. The blue one is the threading results. And then the refinement results is making this green structures, which is 4.4 angstrom. And in this example, okay, before the refinement, we get 40 angstrom RMSD, because actually C and N terminus was flipped, but after the refinement we could find that the order was different, opposite. This is another example. Actually, there was some small errors well, there were some topological errors in the model, but RMSD is 9.3 angstrom structures. So okay, so overall, I think there is the room for structural bioinformatics people to work on the low resolution structural data recently available from EMMAP and EM tomograms. But one thing to note is that the reliability or variability of the structures determined by EMMAP differs. Well, naturally, relative to depending on the resolution. And we develop methods for shape-based search, and then rigid structure fitting, and then main chain tracing method for high-resolution structures more and more becoming available. This is done by Lyman Monroe and Genki Terashi for MAINMAST. And then Charles is a computer science student who is very good in doing the mathematical part of the 3D Zenki descriptors. Shuzi is maintaining the EMMAP EM surfer. And thank you very much.
So, do we have any questions?
I have a question here. Very nice work. I wonder how your MAINMAST methods resilient to random or especially systematic errors in the EM map. Although towards the end you mentioned about the experimental maps, you compare with Rosetta. It wasn't clear to me whether you have evaluated how your method performs in that experimental map setting as compared to the simulated map.
Okay, so well, our methods also have a weakness. Actually, every method has the same weakness, but in the experimental maps, the resolution of local resolutions are different at different positions. And because that our method is tracing the dense regions, if there are no dense regions, if the resolution is bad for the outside of the structures, the method cannot go to that direction. And if we want to do that, we need to do some help from the fragment from PDBe structure and so on. But that's obvious weakness. But that weakness is severer, seems like in Rosetta, because Rosetta needs to assign the fragment.
Okay. Thank you very much.
Okay, I think in the interest of time that we will keep moving on. So thank you very much again.